Hello, my name is Antonio Ramirez. In this poster, I'm presenting our study where we show that putative epigenetic relationships involving intronless genes are distinguished by having an overactivated status after the transition from healthy to breast cancer cells. Therefore, we'd like to highlight their potential role as biomarkers. Intronless genes are very interesting to study because they avoid the processes related to alternative splicing of transcripts. This suggests increased transcriptional fidelity. We hypothesized that intronless genes could play essential roles in the determination of cell phenotype. Then, the disruption in their co-expression patterns might have a causal relation with the disease characters. We collected transcriptomic data for breast cancer adenocarcinoma samples obtained from the TCGA database, having around 1,000 samples for tumor and more than 100 for adjacent normal tissue. This data allows the inference of co-expression networks for two groups of genes. First, intronless genes, and second, multiexonic genes as a control group. This task was performed using Arachne, a mutual information-based tool for gene regulatory network deconvolution. Co-expression was computed for two conditions, healthy and tumor cells. Now, we define the rewiring process associated with breast cancer as follows. Two genes are connected if they co-express in at least one condition. These connections are characterized by the conditions in which they appear: normal tissue, disease, or both of them. Each query gene gains and loses co-expression links in the transition from healthy to disease condition. This fact depicts a rewiring profile, letting us to group the genes by their behavior in the network. Turn-off genes tend to lose connection in tumor cells, in contrast with turn-on genes whose connection in tumor are considerably higher than in healthy cells. Finally, essential genes have almost the same links in both cases. Network rewiring in breast cancer reveals that the intronless genes tend to be in the turn-on class. Some of the genes that better illustrate these results are H2BC8, H2AW, and H1-2. Intronless genes with higher turn-on degree are related to a set of biological processes, including chromatin condensation, that is essential for regulation of cell cycles. Based on the amount of rewiring, we scored the intronless genes as putative biomarkers for disease. This is a first step towards the identification of determinant process for cancer phenotype emergence. The rewiring process in a network captures changes in coordination of gene expression, which probably imply disruption of cellular processes. In the context of cancer, such changes allow cells to survive, but without proper control of cell cycles and phenotype. Our results shows that breast cancer prompts a gain of new links with intronless genes, presumably because these genes are highly involved in the disease progression, so they have a great potential for biomarkers in breast cancer. Furthermore, we remark the current need to increase the number of samples available in public datasets to infer statistically significant co-expression networks for other cancer types. In the future, this analysis could be performed to compare the rewiring patterns across complex malignancies. Thanks to CONACYT for funding under the Ciencia Básica project 254206.